Hi everyone, welcome to the second video of Oogenesis. In this particular video, we will be learning about oogenesis, how the oogonia will undergo mitosis and meiosis to eventually form a mature egg. In our previous video, we were learning about spermatogenesis. So as we run through this video, let's try to also make some comparisons between spermatogenesis and oogenesis. Now, right here in front of me, I have an embryo. This embryo will eventually become a baby girl. We see in the embryo a collection of cells. These cells, they are colored in green, they are primordial germ cells, and these cells are essentially precursors of the oogonia. In males, the primordial germ cells are also the precursors to the spermatogonia. In this particular video, we'll be looking at oogenesis. So let's begin our story by looking at a single primordial germ cell. Now, at the very top in green, this is a primordial germ cell. The primordial germ cell is diploid in nature. And one of the first things it will do is to undergo mitotic division. And when it undergoes mitotic division, we will expect that the resultant cells are also diploid in nature. We call these cells oogonia, or oogonium if you're talking about just one of them. Now these oogonia, like the spermatogonia, will then undergo another round of mitosis to form diploid cells, and these diploid cells, we call them primary oocytes. Now, things that have been happening so far are quite similar to how primary spermatocytes are formed. However, from this point onwards, things start to change up a little bit. These primary oocytes, just like the primary spermatocytes, will then begin and want to go through meiosis 1. Now, it will start meiosis 1, but it will stop at a certain point, meaning to say it does not complete meiosis 1. It stops, in fact, or as we say it arrests at particular phase. And this phase is prophase 1 of meiosis 1. So what we have here are cells, primordial germ cells that have undergone mitosis to become oogonia, which undergo mitosis to form tons of primary oocytes. And these primary oocytes begin meiosis 1, but they stop or arrest at prophase 1 of meiosis 1. And this all happens before birth. Now the next question you may have is then, when does meiosis 1 run to completion? Well, let's say this embryo before birth has all possible primary oocytes that have been arrested at prophase 1 of meiosis 1. And then the baby girl is born. The baby girl will enjoy about 12 years of her life before puberty strikes. Now at puberty, a special hormone is produced. This hormone, we call it the follicles stimulating hormone. And as the name suggests, this hormone helps to stimulate the development of follicles, as we will later on see. But essentially what we can see from here is that prophase 1 of meiosis 1 will no longer be arrested when this hormone is being secreted. And we can continue on with meiosis 1. So let's say meiosis 1 continues on. And meiosis 1 will continue on, and we will form two cells. And these two cells, take note, they are now haploid in nature. Not only that, you can see that they are different in size. Now, although these two cells contain the same number of chromosomes, both haploid, we find that the cell content is a little bit different. One cell is much smaller. We call this smaller cell the first polar body. We call the larger one the secondary oocyte. Now this secondary oocyte is much bigger than the first polar body because cytokinesis did not occur evenly as the primary oocyte was completing meiosis 1. And therefore you get one cell that's bigger, one cell smaller. One received most of the cytoplasm, the other received very little of it, but it still contains chromosomes. Now that the cell has undergone completed meiosis 1, the secondary oocyte will then begin and want to complete meiosis 2. And so it will begin meiosis 2, but again, it will stop at a certain point. It doesn't actually run to completion. It will stop, in particular, at metaphase 2 of meiosis 2. And so if we say it stops or rests at metaphase 2 of meiosis 2, what will the chromosome arrangement look like within the cell? I'm just going to zoom into the cell over here. Let's try and make sense of what this may look like at metaphase 2 of meiosis 2. I'm going to just draw 2 chromosomes to give you an idea. I'm drawing 2 chromosomes over here. They will be aligned at the metaphase plate, and they've halted at metaphase 2 of meiosis 2. This might be what it will look like, but I've just shown you only 2 chromosomes. So it will halt at this phase for a period of time. What will cause metaphase 2 of meiosis 2 to continue running on? Well, we will require then a sperm to fuse. What will happen is that when a sperm fuses with the secondary oocyte that is currently arrested, then meiosis 2 can run to completion. And when meiosis 2 runs to completion, we will end up getting, again, 2 cells that are haploid. One of them, we can now call it the ovum. The other, we refer to as the second polar body. And as you can see, also, the second polar body is much smaller than the ovum because cytokinesis did not occur evenly. Now, if you look to the left, you may be asking what will happen to the first polar body. Well, 2 things. Number one, the first polar body may disintegrate. If it disintegrates, then, well, there's nothing much else to talk about. But if it does not disintegrate, the first polar body can actually also undergo meiosis 2 to completion, and we get 2 extra second polar bodies. And that's if the first polar body does not disintegrate. And so I've just briefly run through oogenesis, but now let's try to make sense of where all these processes take place in relation to the ovary. Now over here I've drawn an ovary with a lot of structures within it. If I were to zoom out, you can see that this ovary is part of the female reproductive system. The female reproductive system consists of 2 ovaries, and we will just focus our story on one of them at the moment. So now let's zoom all the way in and have a look at the story. So now let's start from the top. We mentioned earlier that before birth, the primordial germ cells would have undergone mitosis to form oogonia. And the oogonia will undergo mitosis to form tons of primary spermatocytes that will be arrested, in particular at prophase 1 of meiosis 1. Where can you find all these cells? We will find all these cells in the ovary within structures that we refer to as primary follicles. What does the structure of a primary follicle consist of? Well, let me just quickly show you. I'm going to extract out this primary oocyte over here. I'm going to bring it within the ovary so that we can have a look at the structure. Now, if we look at this single primary oocyte, this primary oocyte won't just exist in the ovary by itself. Instead, we will find, let me zoom in, that surrounding the primary oocyte is a layer of glycoprotein. We call this layer of glycoprotein the zona pellucida. And surrounding the zona pellucida, we find a single layer of cells. We call these cells follicle cells. And this single layer of follicle cells will surround the zona pellucida. And the entire structure we refer to as a primary follicle. And this is what a primary follicle is. it contains a primary oocyte that has been arrested at prophase 1 of meiosis 1. But this primary follicle doesn't just remain this structure the entire lifetime of the female. As we mentioned earlier on, eventually at puberty, the follicle stimulating hormone will be secreted. What's the role of follicle stimulating hormone? Well, it will cause several primary follicles to begin to develop and grow. I'm going to just focus our attention on one primary follicle developing as a result of follicle stimulating hormone. We find that the primary follicle will begin growing and developing, and you see that as it grows and develops, we find that there are a lot more follicle cells, and we also find that an empty space is also developing. We call this empty space the follicular antrum. The follicular antrum consists and contains nutrients that's necessary for the development of the follicle. What do you expect to find within the this larger follicle? We expect to find a secondary oocyte that has already completed meiosis 1. Okay, so we will find a secondary oocyte that has completed meiosis 1. Not only that, if it's completed meiosis 1, we'll expect to find a small polar body, or the first polar body beside it. We call this entire structure the secondary follicle. Now, the secondary follicle can begin and continue to grow and develop as a result of FSH, and it will eventually grow and become really big. You look at this big structure over here, we call it the Graafian follicle. And this Graafian follicle, you can see that the follicular antrum has grown and become much bigger. This is one of the key features of the Graafian follicle. Very large follicular antrum. Not only that, it has a lot more follicle cells. What would you expect to find within a Graafian follicle? You will expect to find a secondary oocyte that has begun meiosis 2 but has been arrested at metaphase 2. So let me just extract out this secondary oocyte that has begun meiosis 2 but has been arrested at metaphase 2. You will also expect to find still the first polar body within the Graafian follicle. And this is essentially what a Graafian follicle would look like. Then this Graafian follicle won't just remain in the ovary, something will happen. And what will happen is this: the Graafian follicle will undergo ovulation. It will undergo ovulation if there is a surge in a particular hormone, a surge in a hormone called LH, or luteinizing hormone. With the luteinizing hormone, we'll find the Graafian follicle undergoing ovulation. Now I'm going to demonstrate to you ovulation right here. Let me just do a little bit of preparation. I'm going to move this Graafian follicle to the edge of the ovary. Here I've got the Graafian follicle here. I'm going to move this Graafian follicle to the edge of the ovary. We said that the luteinizing hormone will cause it to undergo ovulation. Now the ovary will rupture, and when it ruptures, it will then release the secondary oocyte that is arrested at metaphase 2 or meiosis 2, along with some follicle cells. Okay, let me just extract this out. And this is essentially the structure of an egg. We find that the egg doesn't just come out by itself. It also brings along with it the zona pellucida. And also along with it a collection of follicle cells surrounding it. We call this collection of follicle cells now the corona radiata. And here we still have the zona pellucida. And inside we have a secondary oocyte that is arrested at metaphase 2 of meiosis 2. Now, when will metaphase 2 of meiosis 2 continue on to completion? If a sperm fuses with the secondary oocyte. So right now, imagine a sperm has arrived. I'm going to draw a sperm. One of the first things the sperm will do is to push its way through the corona radiata, like push aside all the follicle cells. Eventually, the sperm will reach the zona pellucida. When it reaches the zona pellucida, inside the sperm is the acrosome. Acrosomal reaction will occur such that it will digest the zona pellucida so that it can reach the secondary oocyte. And when it reaches the secondary oocyte, its membrane will fuse with that of the secondary oocyte. So here I'm trying to show a little bit of fusion. I'm going to erase away a little bit of the secondary oocyte and show the fusion over here. Okay, this is a sperm fusing. And as it fuses, you'll find that the nucleus within the sperm that was containing the haploid number of chromosomes will also be able to enter the secondary oocyte. So this is haploid in nature. And when all this begins to happen, metaphase 2 or meiosis 2 will continue on to the next phase, which is anaphase. So here I'm going to erase this all away because I'm going to continue on the process to metaphase. To anaphase. Now you'll find that the chromosomes are being pulled apart. This is anaphase. And after it's gone through anaphase, the chromosomes will then come together and become enveloped in the nucleus. This is telophase beginning. And of course, the cell will then undergo cytokinesis. But remember, the cytokinesis doesn't happen very equally. What we will get is one cell that's bigger than the other. And we'll get one small cell at the side. And this is the second polar body. And the second polar body will contain the chromosomes, haploid number of chromosomes. Now we call this structure a mature ovum or egg. And this egg contains also with it haploid number of chromosomes in the nucleus. And when the nucleus of the ovum and the nucleus of the sperm come together, fuse together, let me just draw a quick fusion. When they fuse together, that's fertilization for you. Let me just use it purple now since they fuse together. Okay, all the chromosomes from the ovum and the sperm are now mingling with each other. Here now we've got something we call a zygote. And this zygote won't remain a zygote. It will undergo a process called mitosis to form a collection of cells called the blastocyst. And this blastocyst will then undergo even more rounds of mitosis to form something we call an embryo. And in a sense, we have come full circle. This embryo is the embryo that we talked about at the very beginning. Over here. Now this embryo will eventually have its own primordial germ cells. The primordial germ cells may go on to develop and become spermatogonia, or it may go on to develop and become oogonia, depending on the gender. And in a sense, the whole cycle repeats. We've come to the very beginning. So I hope these two videos have helped you understand the differences and similarities between spermatogenesis and oogenesis, and also helped you to draw some relations to where exactly all these processes take place. You can follow on in the next series of videos to explore the menstrual cycle and the ovarian cycle, and dive a little bit deeper into the hormones that help to grow and develop the follicles within the ovary. With that, thank you, and I hope you found this video useful.